This is the first lecture on nutrition. Energy intake is balanced with energy output in healthy individuals who maintain a stable body weight. Because different foods contain different proportions of nutrients, appropriate balances must also be maintained among these nutrients so that all segments of the body's metabolic systems can be supplied with the requisite materials. Today, we will cover 3 topics. First, we will look at ATP and nutrients. ATP, or the similar nucleotide, GTP, is used as an energy source for almost all cellular functions. An attribute of ATP that makes it highly valuable as an energy currency is the large quantity of free energy. The amount of energy in each bond, when liberated by decomposition of ATP, is enough to cause almost any step of any chemical reaction in the body to take place, if appropriate energy transfer is achieved. ATP energizes the synthesis of cellular components, such as proteins, glucose, fatty acids, and almost all other organic compounds of the body. In addition, ATP energizes muscle contraction, active transport across membranes, glandular secretion, and nerve conduction. Despite the importance of ATP for the cells, ATP is not the most abundant store of high-energy phosphate bonds in the cell. Phosphocreatine, that also contains high-energy phosphate bonds, is 3 to 8 times more abundant than ATP. This is phosphocreatine. It is derived from arginine and glycine. Phosphocreatine and ADP can be easily converted to ATP and creatine because of the higher energy level of the phosphate bond in phosphocreatine. Therefore, even the slightest use of ATP by the cell will set forth conversion of ADP to ATP as long as phosphocreatine remains. The energy content of food is calculated from the heat released by the total combustion of food in a calorimeter. It is expressed in kilocalories. Energy from fat is 9 kilocalories per gram. Energy from alcohol is 7 kilocalories per gram. Energy from proteins and carbohydrates is 4 kilocalories per gram. We should remember these numbers. For example, a recommended 2,100 kilocalorie diet consists of 58% carbohydrate, 12% protein, and 30% of fat content. How much of each nutrient should one take? To get the amount of carbohydrate, for example, first we need to calculate the energy from carbohydrate. It equals the total calories from the diet times 58%. That's 1,218 calories. This number divided by energy content of carbohydrate, which is 4. This gives us 305 grams for carbohydrate. Using the same method, we can obtain the amounts for protein and fat. Here, I'm going to introduce you 2 methods used to determine metabolic utilization of carbohydrates, fats, and proteins. First method is based on respiratory quotient. Respiratory quotient is the ratio of carbon dioxide production to oxygen utilization. It can be used to estimate usage of carbohydrate and fat. When carbohydrates are metabolized with oxygen, exactly one carbon dioxide molecule is formed for each molecule of oxygen consumed. As a result, the respiratory quotient for carbohydrate is 1. When the fat is oxidized in the cells, an average of 70 carbon dioxide molecules are formed for each 100 molecules of oxygen consumed. So the respiratory quotient of fat averages 0.7. The respiratory quotient of protein is 0.8. The reason why the respiratory quotients for fats and proteins are lower than those for carbohydrates is that fats and proteins contain more hydrogens, which combine with oxygen to form water. As a result, less carbon dioxide is produced. Here are some important findings from study of respiratory quotients. First, immediately after a meal, almost all the food that is metabolized is carbohydrates. As a result, the respiratory quotient at that time approached 1. About 8 to 10 hours after a meal, the body uses fat for energy. As a result, the respiratory quotient at that time is close to 0.7. In untreated diabetes, little carbohydrate can be used because insulin is not there. When diabetes is severe, most of the time respiratory quotient remains close to 0.7. Second method is based on urinary nitrogen secretion to estimate protein metabolism. As we know, the average protein contains about 16% nitrogen. About 90% of this nitrogen is excreted in the urine. The remaining 10% is excreted in the feces. Therefore, the rate of protein breakdown in the body can be estimated by measuring the number of nitrogen in the urine, then adding 10% for the nitrogen excreted in the feces. Or we can do this simply by multiplying urine nitrogen content by 1.1 and then multiplying by 6.25. This will give us the total amount of protein metabolized in gram per day. For example, excretion of 5 grams of nitrogen in the urine in a day means the total nitrogen excretion is 5.5 grams. By multiplying this number by 6.25, this gives us 34.4 grams of total protein breakdown in a day. Under resting conditions, the concentration of ADP in the cells is extremely low. ADP is a major rate-limiting factor for almost all energy metabolism in the body. When the cells become active and ATP is converted into ADP, the concentration of ADP rises. ADP increases the rates of all the reactions for the metabolic release of energy from food. Some energy is derived from food through oxidative metabolism. That energy is called aerobic energy. All macronutrients can be oxidized to cause synthesis of ATP. Some energy is derived from food without utilization of oxygen. That energy is called anaerobic energy. Carbohydrates are the only macronutrients that can be used to provide energy without utilization of oxygen through anaerobic glycolysis. The best source of energy under anaerobic conditions is to store glycogen. For example, during hypoxia, a small amount of oxygen stored in the lung and hemoglobin can keep the metabolic processes functioning for about 2 minutes. After that, anaerobic glycolysis can continue to supply energy for about 1 minute. Glycolysis will be described in lecture N02. Second example, during strenuous bursts of activity, such as 100-meter dash, most of the actual energy required comes from three anaerobic sources. First, ATP already present in the muscle cells. Second, phosphocreatine. Third, energy from anaerobic glycolysis. The maximum amount of ATP in the muscle is about 5 millimolar. ATP can supply energy for about 1 second. Phosphocreatine can supply energy for about 8 seconds. These two together can supply energy for about 10 seconds. Anaerobic glycolysis can supply energy for about 1 minute. After a period of strenuous exercise, a person continues to breathe hard and to consume large amounts of oxygen. This oxygen is used for 5 purposes. First, to reconvert lactic acid to glucose. Second, to reconvert ADP to ATP. Third, to reconvert creatine to phosphocreatine. Fourth, to re-establish normal concentration of oxygen bound with hemoglobin and myoglobin. Fifth, to raise the concentration of oxygen in the lungs to its normal level. This actual consumption of oxygen after exercise is called repaying the oxygen debt. Now, let's move to part 2. We will look at energy intake. When energy intake and energy expenditures are balanced, we can maintain stable body weight. In the hypothalamus, there are 2 centers that regulate food intake. One is feeding center that increases appetite upon stimulation. It is located in the lateral nuclei of the hypothalamus. Another center located in ventral medial nuclei serves as the satiety center. The satiety center inhibits feeding center. In addition, 3 other nuclei also plays a major role in control of appetite. For this class, we will focus on these 2 centers. In summary, food intake is controlled by 2 centers in the hypothalamus. Feeding is modulated by higher center and by signals from gastrointestinal tract. Short-term regulation of food intake is concerned with preventing overeating at each meal. Here are signals that inhibit short-term food intake. First is stretch receptors, found in stomach. When stomach is distended, stretch signals are transmitted by way of vagi to the brainstem and then inhibit feeding center. Appetite for food is reduced. GI hormones, including CCK, peptide YY, glucagon-like peptide, and insulin, also inhibit feeding center. On the other hand, ghrelin is released from stomach and duodenum during fasting. It increases appetite. Long-term regulation of food intake is concerned primarily with maintenance of the normal quantities of energy stores in the body. A rise in blood glucose level increases the activity of satiety center and decreases the activity of feeding center. A rise in blood amino acids or lipids level also have similar effects. Decreased body temperature increases the activity of the feeding center. Leptin, a hormone released by adipocytes, inhibits food intake and decreases energy storage. Here we ask a question: how do signals from gastrointestinal tract and adipocytes affect two centers in the hypothalamus? This slide shows how signals from GI and adipocytes regulate appetite through two centers in the hypothalamus. This is a hypothalamus. Here is an arcuate nucleus, and this is periventricular nucleus. Here we have three neurons. 1, 2, 3. Activation of two neurons in red reduce appetite. Activation of the neuron in green increase appetite. MC4R neuron express MC4 receptor. When the MC4R neuron is stimulated, it reduces appetite. At the same time, it increases energy expenditure by increasing sympathetic activity. POMC neuron expresses POMC and CART. Both are peptides. As we know from biochemistry, POMC can be cleaved to produce many different smaller peptide hormones. One of them is alpha melanocyte stimulating hormone. This hormone binds MC4 receptor on MC4R neuron and stimulates this neuron. MPY neuron secretes MPY and agouti-related peptide. MPY binds receptors on POMC neuron and MC4R neuron and inhibits their activity. Agouti-related peptide blocks the action of alpha melanocyte stimulating hormone. As a result, stimulation of MPY neuron increases appetite. Ghrelin binds as a receptor on MPY neuron and stimulates this neuron, whereby ghrelin increases appetite. CCK, insulin, and leptin bind receptors on MPY neuron and POMC neuron. They inhibit MPY neuron but stimulate POMC neuron. As a result, these hormones reduce appetite. This information is summarized in this slide. This slide shows how leptin plays a role in long-term regulation of food intake. When the amount of adipose tissues increases, Adipocytes produce increased amount of leptin. Leptin has four effects. First, it inhibits MPY neuron and decreases production of MPY and agouti-related peptide. Second, it stimulates POMC neurons. All this decrease food intake. Third, leptin increases sympathetic activity, whereby it increases energy expenditure. Fourth, leptin decreases secretion of insulin. As we know from biochemistry, insulin is an anabolic hormone. Decreased insulin secretion leads to decreased energy storage. This slide summarizes hormones and neurotransmitters that affect feeding. Factors on the left column decreases feeding. They are anorexigenic. Factors on the right column increase feeding. They are orexigenic. Here is a mnemonic that can help us remember these 6 orexigenic factors. No game. It stands for neuropeptide Y. Orexins, ghrelin, agouti-related peptide, melanin concentrating hormone, and endorphins. Now, let's move to part 3. We will look at energy output. Energy output can be divided into several components, including energy used for performing essential metabolic functions of the body, performing various physical activities, digesting, absorbing, and processing food, and maintaining body temperature. An average man who weighs 70 kilograms and lies in bed all the day uses about 1,650 kilocalories of energy per day. Eating and digesting food adds 200 kilocalories. Sitting in the chair without exercise, adding another 150 kilocalories. Therefore, daily energy requirement for a very sedentary man is about 2,000 kilocalories. Basal metabolic rate, or BMR, is the minimum energy expenditure for the body to exist. It accounts for 60% of daily energy expenditure in individuals with moderate activities. BMR can be determined by measuring the rate of oxygen utilization over a given period of time under well-defined conditions. The third largest energy expenditure is for processing food. It accounts for 8% of daily energy use. For people whose diet is mainly carbohydrate, this portion of energy can be lower. For those who consume mainly protein, this portion of energy can be significantly higher. Finally, energy expenditure for non-exercise activity accounts for 7% of daily energy usage. Here is a side note. Shivering provides heat by increasing muscle activity in response to cold stress through motor center for shivering in posterior hypothalamus. Non-shivering thermogenesis is used by infants to increase heat production. In this case, sympathetic stimulation release norepinephrine and epinephrine. These two hormones increase release of free fatty acids from brown fat. Increased level of free fatty acids activate thermogenin. Thermogenin is an uncoupling protein, and it will create a proton leak, which generates heat instead of ATP. This integration with biochemistry. Here is clinical correlation. Obesity is defined as an excess of body fat. A standard marker for obesity is the body mass index, or BMI. BMI is calculated as a person's weight in kilogram divided by the square of the height in meter. A BMI over 30 is considered obese. In general, obesity results from greater energy intake than energy expenditure. Factors that cause obesity include environmental factors and genetic factors. Environmental factors include sedentary lifestyle and abnormal feeding behavior due to many reasons. Genetic factors include mutation of MC4 receptor or mutation on chromosome 15. Leptin or leptin receptor mutation also cause obesity, although it is very rare. A skyrocketing increase in obesity over the past 30 years emphasizes the important role of environmental factors. Here we have a model based on gene-environment interactions. Environmental factors provide a driving force for obesity. Genetic variations enhance susceptibility for weight gain through modification of feeding center or satiety center. Genetic variations also determine preferential size of fat storage. A condition opposite to obesity is inanition. It is extreme weight loss due to inadequate food supply or loss of appetite. Anorexia is a reduction of food intake caused by decreased appetite. Anorexia nervosa is a psychic disorder. People with this condition have a strong desire to be thin. Cachexia is a condition in which increased energy use due to severe metabolic disorder contributes more to weight loss than decreased food intake alone. Patients with cancer, AIDS, or chronic inflammatory disorders typically have cachexia.